So we have pretty close to this.
Yeah.
So stay close to it.
Right. Yeah. First and last time. Flames. Is that for you?
Oh.
Hey, Dayquil, like, man. I wonder if everyone can hear what I'm doing right now.
Probably.
Hello. Yeah, right. Yeah, we're just.
Yeah, it might have been the device that
Yeah, I think we might have heard you go into the bathroom.
I didn't see.
Someone did.
Yeah, it wasn't
Very distinct. It was like the very distinct, like, kind of trinkling, and there's like a flushing sound. Water. Gone in the bathroom before Kristen.
I didn't. I personally go to the bathroom.
No.
Franco was in the hotel room. Yeah.
Oh. That's a deal.
Yeah. Yeah.
Yeah.
Well, you ready to go?
Yeah, I mean, yeah. Seems like most of the people that should be here are here. Wait, where's? Oh, there's. Yeah. Like, is the oh, there's a bond. Yay! A very, very small lab.
That's all.
Yep. Everyone turn their devices off.
All right, you ready?
Yep.
Okay.
Video.
Okay, we're going to go ahead and get started now. So, you all have your snacks, hopefully. We'll have a moment at the end of an hour to restock. Anyways, thanks for coming to Max's presentation of his project. For those of you that don't know, Max completed his Bachelor of Science degree in geology at UC Santa Cruz. And basically, he's my first student here at OSU. He contacted me during the summer that I was moving, having babies and that kind of thing. Well, yeah. And it was a little bit of an epic time. And I didn't quite have a project for him, but we worked through a couple things and settled on a topic that we'll see a couple of aspects of today. But he's been he quickly got involved in many aspects of the college. Let's see. So he was the Winter Social President of 2013. Co-founded Forest Scout, which is a graduate undergrad mentoring program. Helped out on Natural Resources Barbecue, which involves graduate students across the division, geosciences, ag, and then also helped out on the firm. He was on the Fur Barbecue community, and actually competed in timber sports and won first place in the choker race that was qualified in 2013. And he's been really active in the college on the FPC and then on the graduate school council. You know, in my lab, he helped establish my lab here at OSU. Anyways, wrote SOPs, just got things the ball rolling, how to sample come to the lab, and where to go, that kind of thing. And he's been a really active member in my direct community and helped bring new members into the fold and help visiting people find their way around the lab as well as find their way around the valley. Put people up that I visit, that kind of thing. So like we didn't have a project when we started, but we went through probably 5 or so drafts of projects and collected some preliminary data on a few things. But we finally settled on aspects of soil variability in managed forest soils. And so you'll see a couple of those projects today. I want to thank you all for coming out and supporting Max on this. And take it with you here.
Yeah. Oh, thanks, Jeff. Yes, first off, welcome to everyone and coming in so close to the holiday season. Kind of a funky time for defense, but hopefully my committee can give me a pretty sweet Christmas present in about an hour. And I did want to say hi to everybody online. I sent that email to about, it was neat. I started to rate people that I wanted to come tune in, and I got up to about 60 email addresses before I finally was like, I just got to send this email. So hopefully some of those 60 are tuning in from everywhere across the globe. And you will have an opportunity in the chat box to write in. So my dear friend Addie over there is going to be helping you with that. So you can drill me with questions if you want. So yeah, let's just jump right in then. So this is a picture of soil from one of our talented soil faculty members, Jay Noller. And it's funny, people don't really think about soil as often as they think of the above ground components of a forest. And part of the reason is it's probably the most complex thing on Earth. So soil takes aspects of atmospheric science, the biosciences, lithosphere, hydrosphere. Remember all those subjects you had in 10th grade science class? All of those are in soil, yet ironically, you don't ever get soil in 10th grade science class, so that may be one aspect for my future work. For those, soil has a little bit of everything. It's got water, organic matter, it's got matter. When you're walking on soil, I always like to say, in the sense you're walking on air, you're also walking on water, so you know, Jesus' trick wasn't that great. He was just on a really hydric soil, is my theory. Soil is also not just complex, but highly variable. If you think of the word complexity, it also just means variable, and soil is one of the most variable things on Earth. It's soil at different scales, so it has root tips coming in and interacting. There's all kinds of, we have Marcus Cleaver here who works on mineral interactions at the smallest scale, and Jeff also kind of hangs out down here in kind of chemical fun land down there, but there are also plenty of researchers in this room that work at just about every scale here. And there's so many different aspects going on at so many different scales. And I naively, I started as an earth scientist, so I came into soil pretty late and was like, Jeff, this is cool, can I just explore this? And Jeff was like, sure. So here's kind of how the conversation went, and I might need a couple of volunteers. You guys want to come up? This is the lighter version of the conversation, saying, hello, my very accommodating advisor.
Hello, Max.
So I need a project. Hey, can I investigate this causes of spatial variability in soil?
Yeah, that sounds interesting and important. Maybe even we can find you some data.
Okay.
For soil, nutrients, I'm in.
High sample size, high resolution, I'm in. All right, let's do it. That was us 2 years ago. There's some of Scott's data. It looks great. It's fun. As many of you know, I'm a data scientist, so I was just going bonkers with this. This was me last year. Our initial results, trying to look at correlating some of this carbon. For those that don't know, this is about as bad as a data set. It's also kind of a typical master's first run at data. So we're kind of like, uh-oh.
some problems here. I literally went to the drawing board. As Jeff knows, in lab meetings, I'm kind of addicted to whiteboards. So this was me struggling all the time. It was like diagramming stuff. I was like, where does nitrogen actually come from? The atmosphere or the sun? Depends on time scales. And finally, after months and months of banging my head against the wall, sometimes literally, I came up with this sentence, that variability is scale dependent. And Scott's data only existed at one level of scale that he investigated. And I can unpack this sentence. I actually had a whole lecture kind of going into this and realized there's an entire PhD project that revolves around the sentence. It's pretty complex. I won't go into it too much today because I also want to talk about the compositing, or as Jeff like to calls it, the actual results part of my work. But just to give you a little brief tour into this idea of variability. There's this granite. I didn't actually find, I found a granite one day and was looking at it and was like, this granite's really interesting. It's both like kind of the same and kind of different. So homogeneity, which in my terms is going to be something that's made up of the same kind of thing. It's interesting. I talked to a good kind of biochemist friend of mine. She's like, well, homogeneity in chemistry means like the same. I was like, well, where I'm coming from, it can mean mixed but similar. So vote time. We call this rock the extent, and this is a term that I want you to kind of hold on to. This is the extent of our study. We're going to study this granite. We're all going to do science together. Would you think this granite is homogeneous or not? Who votes for homogeneous? Yeah, way to be good. I got a slide just for you on this one. Who votes for heterogeneous? Like, it's crazy, it's all different. Who votes for Max is tricking us and there's more going on here? A little bit of all three. So if I look at the granite and pick a grain size to measure and say, I'm going to look at if this is my scale of observation, or as I like to call it, the grain size of the individual sample, and sample this granite 4 times, it's actually looking kind of pretty similar. I mean, yes, there are different minerals. There's some darker minerals. There's some lighter minerals. But the mix of dark and light is about the same. They're all about the same size. You would actually call this a pretty homogeneous looking mix. So point for Ben. Over at this scale, though, if I zoom in and say, I'm going to look at this really smaller, when I say scale, I say grain size. Remember, the extent hasn't changed, but the grain size has. And say, what is this rock looking like? Well, now there's kind of like darker minerals, there's lighter minerals, and at least in geology, Michael Jackson was wrong, and it does matter if you're black or white. So this one is absolutely heterogeneous. So the answer is that both of you are right, and the trick question is that it entirely depends on the scale that which you look at. So one of my investigations then, one day, I was looking at all this data that Scott sent me, and he gave me this like, there's like oodles of data samples, and I'll show you what those look like in a bit. And I was saying like, Scott, some of this was collected at the point level, some of this has depth in it. What's going on? And I realized that in our conversations, we weren't actually explicitly talking about what scale data was measured at. We were talking about, there's some like bio data that's at like this one kind of quarter plot thing, and the soil data has some size fractions. And I was looking and I was like, this seems to be a problem. It took me a while to realize this is even a problem. And then when I did the literature review, there's not a lot of papers, but the ones that did a good picture. One, this is, unfortunately, there aren't even in soil science about this. I had to cast a pretty wide net in the natural sciences. And a paper from, this is terrestrial geology, said that only 61% of papers will identify both the grain and the extent of the. And if you remember my initial claim, variability is entirely dependent on the grain extent that you measure. I hope I just showed that with the granite. Grain extent, you report a variability measure, kind of means. Also appears geology, which they kind of saying, just read this quote. Clearly identified problems that arise when heterogeneous systems. Surprising that few studies explicitly measure and analyze scale effects. Doing later this talk. And then there's a study this and just kind of overall ecology. There was a classic paper that was Weed's 1980 that was one of the first to really start advocating this terminology. And this group looked at the ISI Web Knowledge, or as we call it now, Google Scholar, and zoom up. And as you go through the years, the terms more and more and more. there's definitely an awareness of scale issues, but in terms of actually investigating, okay, we know scale is important, but why? I tried to look at scale at papers that really put in a multi-scale component, and they're much rarer. We just did a multi-scale study, and then employed with that or just observational. And then to really actually kind of conduct experiments scales and see what happens is this little, and not a lot of literature out there right now. Oh, and also just a quick note on scale itself. When we say scale in terms of explicit terminology, one thing that is happening and might be biasing these results a little bit, the term scale itself has so many different definitions and so many fields. I came from geography. Scale for me was a cartographic ratio. What's the size of the map? 1 inch equals 24 kilometers. There's also statisticians love support as scale. What's their data? They like to think in numbers. They don't care about maps or ecology. They're just like, they're numbers people. And they use their fancy math terms. And so and all of you in this room probably have a slightly different definition of scale in your head. And part of my work that I'm kind of ongoing research for me is just getting us all to hopefully start to converse on a similar level and kind of scale our definition of scale. I just came up with that right now. So, standardizing the data set. Scott was like, oh, I have all this data. Scott was like a data fairy. You just down from the heavens with just oodles and oodles of data. I think I counted, there's actually, if you talk about the numbers in their data set, there's about, well, I took a bunch of scans of numbers. There's like 40,000 points of individual data in Scott's work. And that doesn't even include the crazy FTIR stuff that generates millions of numbers. So I have all these different data sets. And when you look at it from a scale perspective, which is something I didn't do at first, it took me a while, you'll see that some stuff has the size component. Some is, we'll see later, there's block composites where you take an area and then do representative sampling. Some of it has fractions. Some of the data gave me like lat longs and different metrics. It's just kind of all sorts of a mess. And Jeff was like, just like get this to me like by next week and just organize it. That project turned into about a 4 months of off and on trying to get this thing to work. And I presented data some initial results. One problem as a data scientist perspective and looking at how we put scale in terms of a database is that there's just different ways of organizing data in a database. In this example, this is some carbon. So on the left here are or on the right, my left. On the right here, we have different variables. So this is going to be final, there's weights and rock volume percent of soil. And then I did some just basic initial statistical analysis, and I have it for different variables of the soil for different sites. In Oregon, there's a site in Washington. There are different fractions for the soil scientists in the room and different depths that we measured at. All this stuff going on. And then there's all these different variables. And if you go along, I can keep all those variables in the same table. I can break it out by site. So this is all still one table, but now instead of site being over here in its own as just a row, each row gets a site. Now site is defined at the column level. To make it even prettier, we can bust it out and make it 2 different variable sizes, or we can go all the way. And by this point, I can also take all the data and just, rather than focus on every response variable, I'm just looking at, say, coefficient of variation for my variables. I can really focus in on one variable and then put it all in this table. And so I went to Jeff and said, when you say you want one database, I say there's no such thing. Here are 4 different ways to display the same information. And then I said, he was like, okay, what I just want it all in one spot, Max. I would like to have a clear one spot that I can just view everything. And I said, I can do that. Thought I could do it. Took me several months, but breakthrough moment, and I literally ran up to Jeff's office and said, Jeff, I think I have it. Check this out. That wasn't it. This is sorry, I forgot the slide was in here. This is just all the different variables in this data set. Scott did all the stuff that was up to soil chemical. I added topography and spectroscopy. If anyone wants to have fun playing with data, Scott Holub, Weyerhaeuser, over in Springfield, give him a ring. The explicit scales. I was finally looking at this and I said, what we haven't done with all this data is really said what scales exist in this database. There are scales here that I eventually kind of come up with terminology that I have stuff that exists at the point level, at depth. There are sizes and fractions for the soil scientists in the room. And then also, this is the study design from Scott's data. He has individual points, that's what these little dots represent, and then he also grouped them into blocks. And for example, things like the tree measurements, the above ground biomass, they did a representative sample of the block. So that sample for the tree component is actually, it's a sample of a block, so it exists at this block scale. And just to give you a little bit of a demo into different scales and thinking, also teachable soil scientists, this is what we call a representative sample in soil. This is an auger. You stick it into the ground, and then you pick up about like 10 centimeters of soil right here, and that's our sample. So you can imagine if you're standing on a slope, or even in this room, I have the slope of this ground, and the slope can change. It can go up and down. So slope exists at just an XY point. At a given point on Earth, there is a slope. But if I start to dig into the ground and still have topography, the slope kind of disappears. It's the same slope no matter where you go down. Slope doesn't have depth, but soils do. So these different scales going on. And then within that soil sample, I can break it up into different size fractions of that sample, and that's really a scale unto itself. So there are a lot of different scales of analysis going on in soil. And I was starting to run away with this, and Jeff was saying, I was like, oh, Jeff, I want to like investigate what the deal is and like how does this impact everything? And I thought like I had Max's grand theory of scale. And Jeff was like, Max, it's like November. We should probably get it. It's about September, October. Like we should probably actually do a project. So we called it good. I've kind of developed this. I've explicitly defined scales in the database. Really, just actually put these are all the data sets that exist at the point level. That exist at the point level with depth. These are data sets that exist at this level. Put that all in data. Made it all in pretty colors. Everyone likes colors. And this is the product I'm shipping off to Scott. So he thought he was going to get a really neat kind of just method conclusion study. And instead, he was one of the better looking databases for soil that's ever been created. All right. So before getting through scale completely, I just want to take you on a brief tour to get everyone thinking more in terms of the scale at which our universe is. So this is going to be probably a future area of work, having interesting conversations. I don't know where I'm taking this yet. It's still kind of this ongoing thing. But just to get you guys in the moment, kind of start to think in this multi-scale dimensions, kind of my final pitch for you. I have this little video, and we're going to go on a little journey through the universe. And watch out, it's going to get a little dark in here. All right, I'm going to take you through the entire universe in about a minute.
You slowly see the galaxies like dust is what most of space looks like.
Oh, and for those online, I'm sorry, this looks really bad and crappy resolution.
The trip back to the picnic on the lakefront is sped up version, reducing distance to the Earth's surface by 1 power of 10 every 2 seconds. In each 2 seconds,
Here's where we live.
to cover 90% of the remaining distance back to Earth. To great activity and relatively inactive. A rhythm that will continue all the way to our next goal, a proton in the nucleus of a carbon atom beneath the skin in the hand of a sleeping man at the picnic.
Gotta love the 1970s video.
10 to the 9th meters, 10 to the 8th, 7, 6, 5, 4, 3, 2, 1. We are back at our starting point. We slow up at 1 meter, 10 to the 0 power.
All right. So one way to think about that, technically we're studying that guy's hand and said, where was it? At every scale there, that guy's hand was in that box. So scale, it's just a thing that we don't usually think about, partially because we ourselves as humans kind of exist at this like 1 to 3 meter scale. And so just something I'd like you all to kind of take away, and as you go out today, just think about the scales around you. So then, as far as an actual scientific project, that was fun and all, gave me a lot of headaches writing on boards and good drunken nights with the lab mates. But in terms of like, well, okay, how does scale actually impact studies and looking at kind of a method study? I really like analyzing clearly the how we measure. I kind of call myself a meta-analyzer or an analyzer of analysis itself. And there's always been this long-standing thing in soil science called composite sampling, and there's been a debate about whether it's good or bad. There's different ways you can sample individually, or you can mush stuff in a ball. And I'm going to do, for those of you that aren't familiar, we're going to go over composite, what it is. But the idea is that we do this all the time. It's been done since kind of the dawn of soil science. And I asked Jeff one time, like, well, okay, tell me like straight up, what are the advantages and disadvantages? Jeff was kind of like, well, it just saves money. It's good, right? Like, I don't know. I went to the statisticians and they were like, oh no, God, like we don't know what's going on with the numbers. There was clearly some like active debate about whether it's good or not. And I decided to kind of jump right into that. So what composite sampling is. If you take a bunch of soil samples, this is all from, this is Scott's data. We dragged about 1,000 different bags up to OSU. And by the way, for Scott and everyone exactly familiar with this project, these bags some of them were pre-composited, so this is definitely pictures I put together, exactly what happened in the data set. This is an illustrative purpose only. So yeah, to everyone right now, but that's part of what we We hand wave through those fields. We know we're sampling, so we may get average is going to be a little off. We can build an estimate of the average for tree field A that I'm going to get, and an estimate of the sample averages for tree field B. And the goal in my study is if you take a sample from over here, so you're going to sample tree field B, and you're going to get some sample averages. And we want to know that if your sample averages lie way out here, we know that if the tree field B had the same mean as tree field A, they would result in, if it had a different mean, if the tree field A had the same mean, it would come from the same distribution, and you would always get values kind of over here in the 80s and 90s. But if tree field B, like in my example, has a higher mean, and you start to draw samples, sometimes you'll sample the 3 smallest trees, sometimes you'll get more. This is hypothesis testing. If you ever heard the term significance, this is what significance looks like. And the more often that we can get this curve to result in values that land over here, we're pretty confident that the true means in the field is different. That's called power. There's some more complexity that I'm skipping over, but for what I want you guys to take away, power is good. The more times we hit power, the better. If the means are bigger, so say this second field has a higher mean, then this curve moves over on the right, and there's a lot more power in this study. So let's now look at composite sampling. To test composite versus individual sampling, I wanted to see how a t-test for difference in means is going to operate on populations of individuals or composite samples. So I have 5 populations here, A through E, at increasing difference in means. So as you go higher in the alphabet, you're getting a bigger difference in means, and power should go up of your study theoretically. Well, power always goes up as the means increase, but what I was going to investigate was just by how much. Like, let's put some quantitative numbers on there. I also am going to draw from these same, this is just a normal distribution. So my case is just normal. Stick with, we all, the first curve you ever see, we all know it, we love it, it's kind of like our family. I mean, it has its own Gaussian family, but then you went home for Christmas because the other Gaussian fun of it. One of the things that, remember that a composite samples, I call them averages of averages. When you take a composite sample, we know that the extremes, in the composite case, even if I had the 3 largest trees, if I sneak another tree in there, the average of those 4 is going to be kind of closer to the true mean. So the distribution for composites is a little smaller. They're drawing from this population. Downside is when you composite, your number of test samples goes down. So it's a bit of a trade-off here between the increasing variability in the individual case versus the composites, much tighter variability, a lot more certainty in your estimates, but also less samples to test, so less times you're drawing from here. So I have, so in the individual case, then just going back to that terminology, we have individual and composite, we have individual samples from, right, we have collected samples. So we collect either individually or we collect as a composite and put them in a box and mush them together. And then our test will either result in individual tests or composite tests. So in the case above where I had 3 individual samples, I can take 3 individual samples, take 3 tests, got test size of 3. In the composite case, I can take 24 samples, mush them together into 3 groups, take 3 tests, and then the number of collected samples is 24, but the number of tested is still 3. I pitched this to Jeff, and he said, yeah, math is cool, but how does this work in the real world? Like, I'm over here in soil, and we don't have your fancy normal distributions. So he challenged me to come up with some real-world data. I did that. So I have a carbon data set from a Weyerhaeuser study, and I looked at the data set. This is a right-skewed carbon distribution, and I said, okay, I can simulate that, increase the means on this, and kind of do a hypothetical, what if % increase across the board to this whole data set, or a 3 or 5% increase, and then tested that. How do we test? So here are the sampling schemes. Without going too much into it, the key to know is that in both the normal Gaussian case and the real world case, that I tested both individuals against a composite example where the number of collected samples was the same, 50 and 50, or the number of tested samples was the same, 50 and 50. And in the composite case, if you test the same amount of samples, you have more collected samples. I did this for both the normal and the real world. I played around, where the numbers came from, if you're curious, these are just, I played around with it to kind of generate good results. These are all just a little hypothetical first exploration. All right. If I took samples and just kind of randomly did it once and then sampled it and looked at individual and composite, great. I could compare individual and composite samples once, but that's a sample size of n equals 1 for the looking at differences, and that's a bad sample size. Thanks to computers, why do something once when you can make the computer do it 10,000 times? Monte Carlo simulation is this method of repeatedly sampling random data. It sounds fancy, but it really just means like I can keep taking this Gaussian curve and keep running samples, and you can just go in R and say, give me 50 samples, random samples from this curve. You can do it in Yahtzee all the time if you ever play. You say, I have 5 dice, so in this case, n equals 5, and you can just kind of repeatedly roll and roll many different iterations and see what numbers. And if you kept rolling dice, you'd find out that you probably, you most often see 3s and 4s and averages, and it'd be really extreme to get 6 6s. But it can happen, and then you get Yahtzee and you scream and everyone's happy. So to sample the real world data, by the way, since we don't actually, I can't go into a field and sample real data 10,000 times, at least not without a really good NSF proposal. That'd be fun. What you can do is treat this distribution as a random distribution itself, and there's a technique called bootstrapping, where you just sample from the real data repeatedly and use that as a kind of pseudo representation of your population. All right, so what are the results? So this is for the Gaussian curve up here. And on the top, and there are going to be 2 result slides. On the top is the individual case. So this is just typically 50 samples. I sampled, tested 50. And with this graph, it's roughly a graph of power. These are actually p-values, and they relate. I don't really want to go too crazy into the math stats. But generally, the idea here is that there's more power as your p-values get smaller and smaller and smaller. So over here on the A versus E curve, this is doing great. If you're below this blue line, which is roughly the 10% certainty threshold, it's saying that over my 10,000 samples, how many times did I see that that my test said, oh, you can say with confidence that there's a true difference in means. And we actually did a test case where, hey, like there isn't really a difference in means. And fun fact about science, when there's not a true difference in means, thanks to randomization, you can actually get a significant result. So that's one form of error. And then as we go across, on the top here, the difference in means is increasing. And by the time you get to a difference in starting at 0 and 1.5, and means is 0.5 on a normal, you're almost always seeing a significant result. In the composite case, where I way more samples but put the same amount of tests, is clearly out individual sampling. It's more samples collected but the same number of tests, more cost investment on your part if you're doing a sample. You're going to be able to detect a smaller difference in means a lot more often with the same number of test samples. On the case where you composite, where you collect the same number of samples, so collected is now 50 on the top and the bottom, and only test 5 samples, this part I love. The bottom graph here looks almost identical to the top graph. It's saying that if I only collect 50 samples, and in one case just smush them up, admittedly perfect compositing, so there's going to be some talk about that at the end, but in a perfect scenario, you can only test 5 samples, and you're getting about the same power performance out of the t-test as you would in the individual case. Real-world data. Real-world data looks almost exactly the same. Here's the individual case going down as you increase the means. In the case where you collect the same amount but test fewer, performs about identically. In the case where you collect more samples, or in this case, yeah, I varied this up a little bit because it didn't make sense to test 300 or to composite 300 samples. It didn't make, yeah, this is a little bit, I tricked myself a little bit. It didn't make you 25 individual samples and test 25 because I was trying to go for a more realistic scenario that Scott could use in his work. So I said this third one is what happens if Scott had gone and collected twice the number of soils but only tested the same amount. And in the case where you in collection size but test the same amount, 2 versus 3, collecting more than the same amount really helps improve performance in your model or performance in your t-test. What are the general takeaways here? T-test, it's a lot better to, if you only have a choice between collecting field samples or testing more samples, it seemed to indicate collect more field samples. And in my head, I like to think of that samples are the information, right? That's what you're really sampling. If you have the same number of samples, and in one case you composite or don't, in a sense, you still have the same information there. You still have 50 samples in each case. And the fact that you average them to test them doesn't kind of degrades the information a little bit, but doesn't lose it. So it's kind of a rough information theory. So yeah, compositing does decrease lab costs. I was pretty shocked by that result that in this normalized case, if you collect 50 and test 50 versus collecting 50 and testing 5, that's 45 less samples that you've tested. And you got about the same amount of results in your power. So heck yeah, it can decrease lab costs. But there is the downside, and this is where I like to challenge people to think explicitly on their scale. It's going to change the analysis unit. T-tests are all about the overall average of the field. And if that's all you want to know, did carbon in my whole field go up? That's great. The minute you start asking questions like, is one part of my field have more carbon? Now you're talking about different grain sizes, and you got to be real, in my mind, about looking at what your grain size that relates ecologically to what you're trying to test. So on the whole, I want to say this was a rough rule. If you just, if you took all 300 samples from your field, mushed the entire 300 set together, made one composite, tested that, you need a little bit of it. An n equals 1 is always bad in stats, almost no matter what. So, try and use at least 5 samples per composite. Try and, here I tested 5 samples. If I simulations really to degrade, if I only had 2 test samples, but if I got the test sample up to 5, it worked pretty well. And I didn't really look at the case where if I can only samples, be done. This is still just kind of preliminary work. I haven't found the limits, but rough rules of thumb, yeah, compositing can do some great stuff. Also, a stats professor said, that's all great. You've done these pretty samples, but come on, like, is this actually happening in the math? So I went and you guys with it, but if you want stats with me, I love a pint and a napkin and yeah, just busting out some equations. Lingering questions. Data is spatially autocorrelated, so if there's a field, how does that affect this? We know compositing isn't perfect. And then Jeff had a really interesting note that, if you take volume, so we almost always composite by volume, right? So I'm going to take 5 samples that are at this volume. But sometimes what we measure is the mass of the sample inside. So we talk about how many grams of carbon are in there. So if you have different densities of material and you composite by volume, but measure by mass, then you're getting some bias because the samples are going to be overrepresented in your data. So there's still some questions about compositing, but like I said, I haven't found it in the literature yet, and this is just a first stab at really starting to bunker down and like actually answer these questions. I think simulation modeling is a great way to go about it. Okay, we have now made it. I'd like to thank the College of Forestry. We're right here. This is how much of Oregon is forest. I had a lot of great help. We have one of the most amazing technical support teams, I think, anywhere in OSU. Gary helped me set all this up. The Forestry help desk, if your data gets lost or anything, you can whine and complain, and they'll just say, that's cool, we can handle it. Also, we have a great programmer, Aaron Muldoon, who does our R for us and helps us with coding. Jerry Moore was a great inspiration. I came from a geography background and work a lot with GIS, and a lot of people don't know what the heck I'm talking about when I say, oh, my raster got corrupted into the different projection, and they're all like, talking about soil nutrients. Our firm administrative team, Chelsea, and Angela used to be here, but then she moved because Wood Science stole her. That entire office is always awesome and helping with all the technical aspects. I also want to show, professors are really busy, really busy, and so many professors have still opened their door and said, yeah, I'll chat for you for half an hour about your crazy brain scale theories or your problems. And I'm kind of actually floored when I started to put this list together. There are some professors who are busy and said no or weren't interested, and that happens, but so many here also said yes and helped me out. And I just really wanted to emphasize that they're very awesome people, and don't be afraid to go up to one of those crazy smarties and say, hey, here's an idea. Also, all the college organizations that Jeff went through. We have 2 collaborators in the room, Scott over at Weyerhaeuser, and then his whole soil team below him, which was Nate, and I'm not sure if you have any other buddies now. went on some fun trips into the forest a lot of times, a lot of hiking in the woods. And then there was an entire set of EPA data that I didn't really get to mention here. It was another area we went down, and one thing that happens in science a lot is you find a bunch of data and you don't have any results, and that's fun. So Mark was awesome helping with that work, and the data's ready. It's in the database too. So there's actually, I added an entire spectroscopic set to Scott's data, and I can either do it, or hopefully with the database as it is now, we can hand it off to Jeff's next master's student and say, your data's good to go, give me some results. Then of course, there's the friends and family slide. One thing about working in the College of Forestry and at OSU is that you live in Oregon, and you get to go to so many cool spots. So when I wasn't in the lab crushing samples and compositing or doing lab data, I've been kind of all across the state. There's still some gaps out here in the Steens and kind of the lower west, but yeah, I've had so many wonderful friends from so many different walks of life. It's been amazing. And you know, mom too, she's kind of down here. She hates every picture. Hi, mom. But yeah, thank you to all. And with that, thank you for coming out. And for those of you online, if you'd like to ask any questions, Addie is going to be able to help you out there and get in on this talk, and or general audience.
Okay, so
Yeah.
probably leave about 15 minutes for questions.
Yeah, Mark, hit it.
So, Max, your general, it seemed like you almost had a rule of thumb there that if I use 10% of my individual samples, that was a pretty good composite. So
Yeah.
from 50 to 5, is that what you were saying?
Yeah, so I did, I actually do have a slide. I started some investigations into what level of compositing that you really start to get some drift in your results. Thing that happens when you composite is that out here, here are my 300 individual samples. They will always have the same average. As you start to composite, sometimes you'll get higher samples or lower, like, you know, the 3 high ones may get bunched together, and that's going to drift you farther from the mean. Or if your composites all, like the high one and the low one, wind up in the same composite and get composited together, they're going to get much closer to the average. So here this is closer to the true average versus the individual number, and how that affects the t-test. And compositing does start to drift. So the fewer samples you have per composite, like over here, you get some really nasty drift in the variation. But as you're back in here, these are all t-test p-values. And so this is the individual. So this particular example, the individual had a p-value of 0.3, can only ever have a p-value of 0.3. The composite tends to drift around that a little bit. And so I haven't explored kind of the limits of my rule of thumb. Still just kind of preliminary work. It's definitely something I want to investigate in the future. But I do think that in general, compositing will hold to similar values as individuals. And you can get, so you watch this drift, your value is about, you know, this is 0.3, and as you start to put compositing by 20 and like only 10 samples or 5, about 10 and 15 samples in real world data, you're still doing really well as compared to the 300 individual. So you're, you know, drop 300 to 30, that 10% rule would put you about here. So it's still doing pretty darn well, but there's going to be a risk of increased drift on the p-value, but it shouldn't be too bad. And again, your individual, right, this is only one individual sample, so this individual sample already has some error in it, so it's not like the individual is the perfect example. Yeah, Addie, you got a question for? Yeah, so and I also forgot that online you guys can't actually hear the questions in the room, so my bad for not repeating. Mark was asking about my rule of thumb for compositing, which is what that last question was, and then my friend Tess was asking about some kind of general implications for compositing versus individual samples. Let me go back to that. Where are we? Where is that beautiful slide? All right. So Tess was asking about some kind of general implications for the forest industry about composite versus individual samples. And what I'm increasingly, I have this term that's been floating around my head that I like to call your kind of ecologically relevant scales of analysis. If you're a forester interested in looking at how each tree uptakes nutrients, and you want to know how does one tree see nutrients differently than another tree, if you're at the tree level, then down here at the, so here's my individual sample block. If if we say that like this box represents how much a tree can see, like each individual tree can only see the nutrient, and I only take one sample out of there, I've only taken one sample out of what that tree can see. And we know that soil varies highly across the site. So each individual sample is only really representative, is a sample of one for what that tree sees. If you composite sample, well, now I have 7 samples and I can kind of get more of an average, and I can start to get some estimates for what smaller scales are seeing. So compositing helps you get better estimates of averages for your grain size units. Yeah, Steve over there.
So the Christmas tree slide
Yeah.
is really helpful because when Fisher developed his fundamental ideas of statistics and agronomy,
Right.
he based it on crops.
Yeah.
So we have this phrase, independent, identically distributed units.
Right.
Is that thing that we sample.
Yeah.
Those trees are nice. I mean, there may be some spatial correlation, but they're nice independent units.
Right.
Samples. When I go to take a soil sample, I can choose a core that's that big, or that big, or that big, or that
Right.
big. There is no unit.
Yeah. So
How do you wrestle with that fundamentally in the statistics of soil sampling method?
Right.
The individual is not like that. It's not an individual thing.
Yeah, so that goes back to some of my initial kind of work with the this slide. Come on. I wrestled with that a lot. That's what a lot of the diagrams on here were. You can see there's one right there with looking at different plots and units. So going all the way back to here. Steve was asking about kind of the fundamental unit of sampling. That, for example, with a tree, you have a tree. A tree is a tree. And I know there are like 33 trees on my soil site, and I can go get a sample and build on my stats tests. With soil, there's no such thing as a soil. There's actually not even a standard definition of what soil is, because some people include the forest layer of duff on top of the soil. Like, we don't even have a soil. So there is really no fundamental sample unit, and it's because this is what my arrow that I call variable variability. So as you increase and decrease scales in soil, there's just all this variance at every grain size. And that's something somewhat unique to soil. In the talk that I showed, that Powers of 10 video, there's a really key line where, if you remember from that video I showed, that at the point, sometimes there was the Milky Way and there was a lot of variation, and there were all these gaps where there wasn't much variation. And one of the works I'd like to pursue is seeing where in soil those gaps are, and where is variation in less, and we can expect things to be roughly the same at the extent and grain size that we know. And until we can do that, there's some pretty nasty challenges with studying soil variability. The nice thing about, one of the nice things for us is that what I showed from my compositing is, irregardless of the grain size, if you're looking at an extent, and see, so if I'm talking about this whole stand of trees, and I want to just get the average carbon in that stand, measure at the composite level, I can measure at the interval, doesn't matter, average for that stand. If I want to start talking about variations in the stand, though, different grain sizes have different variation. Variability is scale dependent. So I think to kind of all of that, rolling back to answer your question, studies that do difference in means tests, for example, like fertilizing one field versus another field, you get some replicates. I think those are doing a pretty great job at answering the question they're designed for. But other studies that might have a little bit more question about what the scale, what the ecological scale of the thing they're really trying to bear, like if we're talking about nutrient flux, things that happen at really, really smaller scales. When we talk about nutrient flux on the stand level, nutrient flux through a whole stand kind of doesn't make sense, because we know that whether it's like preferential flow pathways, there are all these things that are happening at smaller scales. And upscaling that to a stand level is kind of arbitrary, and definitely is an area of future work. Yep, James. Yeah. Right. Yeah, James was asking about, one of the things in Steve's question was talking about sample independence, and the idea that in statistics we have called independently identically distributed, which, sorry, I'm jumping around slides a lot here, spastic for everyone watching online. I want to get back to that slide. Go to review. All right, here we go. So this idea of independent and identical. So if you're talking about different samples here, you'll note it's a bit small, but here's my distribution for one sample box, and it's 10 meter by 10 meter by 10 meter. Statistics sort of relies on the idea that if I'm taking samples from across this field, one of the classic assumptions of sampling is that every box has this little curve, and if I just do different samples along that curve, everything will be fine and dandy. know in ecology is there's really no such thing as independentness in ecology. Everywhere is different in the world. There's microclimates. One tree grows a little more, one tree grows a little less. Technically, almost any ecological study at some level violates this idea of independence. And we just kind of, the nice thing is we have shown work that, violating these assumptions still, you can still get saved a little bit. Central limit theorem is one of the coolest things that exists on the planet, because it says no matter what your distribution is, we know the distribution of the curve for the average, and then we can start to play around with stats on that. So anytime you start talking, you're saving the theorem. Anytime you start talking about individual variables, there's no central limit theorem there, and then you're kind of just at the whim of this massive beast. Any other questions? Mark?
So, tell us.
Ooh, yeah. So Mark wants the perfect composite. Well, the word perfect's always fun. This is one, this study, we came in a little late. I've actually only been working on this composite issue for the last 3 or 4 months. And for my samples, I decided to just kind of eschew the idea that there's variation and lab error in composite samples. Like I was talking about with Jeff, there's that mass and volume issue. I mean, really what you'd want to do, what I would do to tackle that question is look at, 8 individual samples and then test them individually and hopefully non-destructively, and then composite, and then test the composite and look at the average of 8 individual samples and the composite, and if they match perfectly, that's what the composite sample is supposed to do. It's supposed to tell you the average. And if they don't, they don't. And I'm not going to make any wide assumptions about that one way or the other. I can tell you, good news for Scott, that Scott had some individual data and some composite data. They were separated, so they weren't the exact same sample, but Scott did some measurements on carbon and nitrogen, both individually and composite, and the numbers were only off on, I should have had that slide up here, but they were only off by about 1% on their carbon estimate, so, and often much tighter than that. So at least in Scott's data set, looks like compositing was doing just about as well at estimating the site mean as individual. And remember, this is the individual composite versus, this is one composite sample. This is how well is one composite sample doing versus the individuals. The other key is you don't have one composite, you have 5 or 6 or 7 composites. So even if the composite is missing, you're off on your average a little bit, and one composite's a little high versus the individuals and one's a little low, assuming that that's random and not biased by, there might be some bias there where it's always overestimating, but if it's just randomly erroneous, then when you go back to the mean, central limit theorem, thank you, the estimate of the mean is still going to be pretty good. All right.
Right, well, if there's not any other questions,
Okay.
and yes, I guess I'll invite you to enjoy Max's service.
Woo, first OSU defense. There is no bar in which to judge whether I did good or bad. All right.
I think you probably had a pretty high bar for this. It's like a really nice friend.
Yeah. Okay. Well, next one out.
How you feeling? How you feeling?
Doing good.
Yeah?
Yeah.
For
Yeah.
about an hour or so.
Yeah. Pretty typical. Yeah. Yeah, they went a little long, but so be it.
Drawbacks.
Yeah.
I like the analysis you did. We're kind of the kind of theoretical effects of
Yeah.